My name is Dr. Elena Kuznetsova, and I'm a molecular biologist with 15 years of experience in genetic research across Europe and the United States. I've spent my career working with some of the most advanced gene editing technologies in the world, including CRISPR-Cas9, the revolutionary tool that's been hailed as humanity's greatest scientific breakthrough. I started in Moscow in the early 2000s, analyzing DNA sequences for pharmaceutical companies, then moved to Boston in 2010 to work with cutting-edge CRISPR applications. I thought I understood the science, the ethics, and the potential of genetic engineering. 6 months ago, in February 2025, a routine research project led me down a path that changed everything I believed about modern genetics. What I discovered wasn't just disturbing, it was terrifying, because I found evidence that CRISPR, the technology we use to cure diseases and edit genes with surgical precision, has roots that trace directly back to Soviet bioweapons programs from the 1960s. Programs that were designed to create what their scientists literally called genetic sorcery. I'm sharing this story because I believe people need to understand what's really happening in genetic research laboratories around the world. The line between healing and harming, between science and biological warfare, has become dangerously blurred, and the implications for humanity's future are more serious than most people realize. It started with what seemed like a simple literature review for an ethics compliance report. I was working late in my home office in Cambridge, Massachusetts. Snow was piling up outside my window, and I had my dual monitors filled with genetic sequences, the digital code that makes up all life on Earth. I was designing guide RNAs for a CRISPR application to treat hereditary disorders, essentially programming molecular scissors to cut out faulty genes and replace them with healthy ones. For those unfamiliar with CRISPR, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a natural defense system found in bacteria that scientists discovered in the late 1980s. But it wasn't until 2012 that researchers like Jennifer Doudna and Emmanuel Charpentier figured out how to harness it for precise gene editing. Their work earned them the 2020 Nobel Prize, and today CRISPR is used everywhere, treating sickle cell disease, creating pest-resistant crops, and potentially developing therapies for cancer and Alzheimer's. But as I was reviewing scientific literature to ensure our research met ethical guidelines, something caught my attention. I was cross-referencing modern CRISPR papers with historical documents about genetic research, and I stumbled across declassified Soviet files in the CIA's online reading room. What I found there made my hands literally start trembling. Have you ever had that moment when you're reading something and your brain keeps telling you that what you're seeing can't be real? That's exactly what happened to me when I opened those Soviet bioweapons documents. The files detailed the Soviet Union's massive biological weapons program called Biopreparat, which began in the 1920s but exploded in scale during the 1960s under Leonid Brezhnev. The USSR operated over 50 secretive laboratories, employing tens of thousands of scientists to engineer deadly pathogens like anthrax, plague, and smallpox. But this wasn't just about stockpiling germs. It was a full-scale offensive program that completely violated the 1972 Biological Weapons Convention, which the Soviets signed but completely ignored. What made my blood run cold was their pioneering work in genetic weapons. In the 1960s, after Stalin's pseudoscientific policies that had suppressed real genetic research were finally abandoned, Soviet biologists threw themselves into genetics with a vengeance. Their goal wasn't just to create superbugs resistant to antibiotics or vaccines. They wanted to develop pathogens that could target specific ethnic groups or even individual people. Think about that for a moment. They were trying to create biological weapons that could be programmed to attack people based on their genetic code. Essentially, they were developing what they themselves called genetic curses that could silently alter DNA, causing diseases that would look like natural hereditary conditions. One document I found described something called Project Chimera, a classified program at facilities like the Vector Institute in Novosibirsk. They were engineering hybrid viruses that could insert harmful genes into human cells, creating delayed-onset diseases or sterility that would be completely undetectable as bioweapons. The victims would simply appear to have bad genetics or environmental health problems. Ken Allebeck, a high-ranking scientist who defected from Biopreparat in 1992, wrote about this in his book Biohazard. He described how Soviet scientists developed what they called invisible anthrax by inserting foreign genes into the anthrax bacteria, making it completely resistant to vaccines and treatments. This was crude genetic engineering by today's standards, but it was laying the groundwork for exactly what CRISPR does so effortlessly now: precise genetic insertion and deletion. As I read through more declassified files, a terrifying pattern began to emerge. The core mechanism that CRISPR uses, Cas9 enzymes that cut DNA at precise locations, was remarkably similar to techniques Soviet scientists were developing in the 1960s using bacteriophages, which are viruses that infect bacteria. I found myself working 16-hour days, barely sleeping, completely consumed by what I was discovering. My wife kept asking me what was wrong. But how do you explain that you found evidence suggesting one of humanity's greatest medical breakthroughs might have been accelerated by knowledge from Soviet biological weapons programs? The breakthrough moment came when I realized the timing. CRISPR's bacterial origins were first identified in 1987 by Japanese scientist Yoshizumi Ishino, but the technology didn't explode into practical applications until 2012 and 2013. Just as Jennifer Doudna's lab demonstrated how to program Cas9 for precise cuts. But what happened between 1991 and 2012 that could have accelerated this research? The Soviet Union collapsed in 1991, and when it did, scientists like Allebeck and Vladimir Pasechnik, who had been working on genetic bioweapons for decades, defected to the West, bringing volumes of research data with them. Vladimir Pasechnik, who defected in 1989, revealed that the Soviets had weaponized over 50 different biological agents. But more importantly, he exposed how Biopreparat's ferment operation was specifically focused on genetically modified pathogens, viruses that could literally rewrite human DNA. They were creating biological weapons that could spread silently through populations, causing symptoms that would look exactly like natural disease outbreaks. When I cross-referenced the dates, everything clicked into place in a way that made me physically sick. Pasechnik defected in '89. Allebeck defected in '92. Both brought detailed research on genetic modification techniques to Western intelligence agencies. And by the mid-90s, research into bacterial defense systems like CRISPR suddenly accelerated dramatically in American and European laboratories. Was this just coincidence? Or had decades of Soviet research into genetic weapons provided the foundation for what we now call precision gene editing? The more I researched, the more connections I found. The Soviet Bioweapons program had conducted realistic field tests of genetic weapons on Vozrozhdeniya Island in the Aral Sea. They would release modified bacteria into the environment to see how they mutated and spread. Accidents were frequent and deadly. A 1971 smallpox outbreak in the nearby city of Arals killed 3 people and was directly traced to these field tests. Today, CRISPR enables something called gene drives, self-spreading genetic edits that can propagate through entire populations. Scientists use this to engineer mosquitoes that can't transmit malaria, which sounds wonderful. But the same technology could theoretically be weaponized to target human genetic markers, causing infertility or disease susceptibility in specific ethnic groups. When you think about it, haven't you noticed how quickly CRISPR technology advanced? From a curious bacterial immune system discovered in the '80s to a precise gene editing tool by 2012. That's remarkably fast for such a complex technology. Unless, of course, scientists already had decades of theoretical and practical knowledge about genetic modification from other sources. The ethical concerns around CRISPR have been exploding since its development. In 2018, Chinese scientist He Jinkui shocked the world by editing human embryos to make them resistant to HIV, creating the first CRISPR babies. The international scientific community condemned this as reckless and unethical, but the technology had already proven it could be done. Military applications followed quickly. DARPA, the Pentagon's advanced research agency, launched something called the Insect Allies Program, using CRISPR-edited insects for what they claim is crop protection. Critics have pointed out that this is essentially a bioweapon delivery system that could be turned against human populations. By 2016, U.S. Intelligence Director James Clapper officially listed genome editing as a weapons of mass destruction threat. Think about that. The same technology we use to cure genetic diseases is now classified as a potential WMD by U.S. intelligence agencies. But here's where the story gets even more disturbing. I started finding evidence that the connection between Soviet bioweapons research and modern CRISPR applications isn't just historical, it's ongoing. Russia inherited most of the Soviet Union's biological research infrastructure, including the Vector Institute, where much of the original genetic weapons research was conducted. In 2024, Russia significantly expanded its Biodefense programs, with multiple intelligence agencies accusing them of continuing weapons research under the cover of defensive research. China has been even more open about military applications of gene editing. The People's Liberation Army has invested heavily in what they call enhanced soldier programs, using CRISPR to edit genes for increased physical endurance, particularly for high-altitude operations. They're literally creating genetically enhanced super soldiers. A 2023 Johns Hopkins University exercise simulated a CRISPR-based bioweapon attack and estimated it could kill over 150 million people if deployed effectively. The scenario involved a genetically modified virus that could spread through airborne transmission while targeting specific genetic markers found in certain populations. When you look at recent disease outbreaks around the world, some patterns start to seem suspicious. Have you noticed how some new viral variants seem to affect certain populations differently than others? How some genetic disorders appear to be increasing in frequency in specific geographic regions? I'm not saying these are definitely the result of biological weapons, but the technology to create such targeted effects absolutely exists now. I decided I needed to dig deeper into who was funding and directing modern CRISPR research. What I found was a complex network of government agencies, military contractors, and private corporations, many of which had connections to former Soviet scientists or their research data. That's when things started getting dangerous for me personally, not in some Hollywood movie way, but in the subtle, bureaucratic manner that actually characterizes how sensitive research is controlled. My laboratory access was suddenly restricted pending what they called a routine security review. Funding for projects involving what they termed dual-use genetic technologies began disappearing. Colleagues started avoiding me, claiming they were too busy to collaborate on research that had been routine just months earlier. I reached out through secure channels to a former Soviet scientist living in exile in Canada. He agreed to speak with me anonymously, and what he told me confirmed my worst fears. In the 1960s, he said, we called it genetic sorcery because it seemed like black magic, altering the fundamental code of life itself. We thought we were developing the ultimate weapon, something that could eliminate enemies without them ever knowing they were under attack. Now, with CRISPR, that science fiction has become reality. But the military applications never stopped being developed. They just moved into more sophisticated laboratories with better cover stories. When you think about folklore and mythology from cultures around the world, curses and hexes that could cause illness, madness, or death in family lines, what if those weren't just superstition? What if ancient practitioners actually discovered ways to alter human genetics using natural compounds? The Soviets essentially formalized this into systematic science, developing viruses that could insert what they literally called genetic curses, causing delayed onset cancers, neurological disorders, or reproductive problems that would be completely undetectable as weapons. CRISPR has perfected this concept. We can now edit human embryos to enhance specific traits, increased muscle mass, disease resistance, improved cognitive function. But these same tools could be used to insert genetic time bombs, programming future health problems, or even controlling behavior through neurochemical modifications. Recent developments have made this even more concerning. In late 2024, several pharmaceutical companies began incorporating CRISPR-based therapies into routine medical treatments. Gene therapies delivered through modified viruses are now being used to treat various conditions, from genetic disorders to certain types of cancer. The delivery mechanisms are remarkably similar to what Soviet scientists were developing 50 years ago for biological weapons. I started tracking the movement of scientists, funding, and research data between countries. What I found was a global network of genetic research that seemed to operate with very little oversight or public awareness. Former Soviet scientists, many now in their 70s and 80s, were serving as consultants for research programs in China, North Korea, Iran, and even some NATO countries. The technology transfer wasn't just about scientific knowledge. It included practical experience in developing genetic modifications that could evade detection, survive in different environmental conditions, and target specific genetic markers found in different populations. But the most disturbing discovery came when I found evidence of testing. Not the kind of dramatic human experimentation you might imagine from movies, but subtle, long-term studies hidden within legitimate medical research. Population genetics studies that seem designed to map genetic vulnerabilities. Clinical trials for gene therapies that included unusual control groups and follow-up protocols. Vaccine research programs that incorporated genetic modification techniques originally developed for biological weapons. Have you noticed any unusual health patterns in your family or community over the past few years? Genetic disorders appearing where they hadn't existed before? Unusual clusters of specific diseases in certain geographic areas? Changes in fertility rates that don't match demographic predictions? I'm not suggesting that every genetic therapy or medical treatment involves biological weapons technology, but the capability to hide such programs within legitimate medical research absolutely exists, and the historical precedent for doing exactly that goes back decades. The turning point for me came in early August 2025, when I received an encrypted message through a secure communication system I used for sensitive research collaboration. The message was simple. Dr. Kuznetsova, your research into historical bioweapons programs has been noted. We suggest you focus your expertise on less sensitive applications of genetic technology. Some doors are better left unopened. The message wasn't threatening in an obvious way, but the implication was clear. I had wandered into territory that someone wanted to keep off limits. Within a week, my security clearance was suspended pending what they called a routine review of foreign contacts. My access to certain research databases was restricted. Most telling, funding for my current projects was temporarily suspended while my research proposal underwent additional ethical review. I realized I needed to document what I had discovered before my access to information was completely cut off. I spent several weeks compiling evidence, cross-referencing documents, and organizing my findings into a coherent narrative. The picture that emerged was even more disturbing than I had initially realized. Modern CRISPR technology isn't just inspired by Soviet bioweapons research. It appears to be a direct continuation of programs that were supposedly discontinued decades ago. The same scientists, the same institutions, the same strategic objectives, all hidden beneath the legitimate veneer of medical research and therapeutic development. What we're seeing now in genetic medicine, the ability to precisely edit human DNA, to target specific genetic markers, to create heritable changes that will be passed to future generations. This is exactly what Soviet bioweapon scientists were trying to achieve in the 1960s and 70s. They just didn't have the technology to do it effectively at the time. Now they do. And the implications for human society, for individual privacy, for the future of human genetic diversity are staggering. We're not just talking about medical breakthroughs. We're talking about the potential for genetic control on a species-wide level. I want to be clear about something important. I'm not opposed to genetic medicine or CRISPR technology. These tools have incredible potential to eliminate genetic diseases, extend human lifespan, and improve quality of life for millions of people. But we need to understand the full history and current applications of this technology, including its potential for misuse. The same precision that allows CRISPR to cure sickle cell disease could be used to insert genetic modifications that cause delayed health problems, reduce fertility, or even alter behavior through neurochemical pathways. The same delivery systems that bring therapeutic genes to patients could carry genetic modifications designed for population control or biological warfare. And unlike traditional biological weapons, genetic weapons could be designed to remain dormant for years or even decades before activating. They could be programmed to affect only certain genetic lineages, making them appear to be natural genetic disorders rather than deliberate attacks. Most significantly, they could create heritable changes that would affect not just the targeted individuals, but their children and grandchildren. This isn't science fiction anymore. The technology exists. The knowledge exists. The historical precedent for developing and deploying such weapons exists. The question isn't whether genetic weapons are possible. It's whether they're already being developed and potentially used. I'm sharing this information because I believe people have the right to know what's happening in genetic research laboratories around the world. The decisions being made about human genetic modification will affect every person on Earth and every generation that follows us. These decisions are too important to be made in secret by governments, military agencies, and corporations without public knowledge or input. When you read about new genetic therapies, breakthrough treatments for genetic diseases, or advances in personalized medicine, I want you to ask questions. Who developed this technology? Where did the research funding come from? What other applications might this technology have? Are there adequate safeguards to prevent misuse? Most importantly, pay attention to patterns. Are certain populations being targeted for genetic research more than others? Are new genetic disorders appearing in unusual geographic clusters? Are there changes in birth rates, disease patterns, or life expectancy that don't match conventional explanations? I can't prove that genetic weapons are currently being deployed against human populations, but I can prove that the technology to create such weapons exists, that programs to develop them have existed for decades, and that the same scientists and institutions involved in historical bioweapons research are now at the forefront of modern genetic medicine. The line between healing and harming, between medicine and warfare, between therapeutic genetic modification and genetic control, has become dangerously blurred. And once genetic modifications are released into human populations, there's no way to recall them. They become part of our species forever. What do you think about these discoveries? Have you noticed any unusual health patterns in your community that might be related to genetic factors? Do you think there's adequate oversight of genetic research programs? Because once you understand the full capabilities of modern genetic technology, it becomes impossible to view recent developments in medicine and public health the same way. The power to rewrite the human genetic code is the ultimate form of control, and history shows us that those who possess such power won't hesitate to use it, regardless of the consequences for humanity as a whole.